Good morning, class. I am your teacher, Charles, and before we start our discussion, may I request everybody to please stand and let us pray. Okay, thank you. Before you take your seats, kindly pick up some pieces of paper under your chairs and arrange it properly. Okay, thank you. Before we start our proper discussion, let me present to you our classroom standard. So these are our classroom standards. So everyone please read. Okay, thank you. Our topic for today is all about recombinant DNA and genetic engineering. So these are our learning outcomes. Okay, please read Rosemary number 1. Okay, thank you. Number 2, Jikimaya. Okay, thank you. So this is this learning outcomes we need to attain after our discussion. So, what is biotechnology? Okay, Joel, please stand. Thank you. So, biotechnology is the use of biology to develop new products, methods, and organisms intended to improve human health and society. So what is recombinant DNA? The recombinant DNA is a method of joining two or more DNA molecules to create a hybrid. So next, these are the principles of biotechnology. The inception of biotechnology was initiated with two technologies. These are the chemical engineering and genetic engineering. So chemical engineering is a branch of engineering concerned with the design and operation industrial chemical plants. Number 2 is it involves the production and manufacturing of products through chemical processes. Number 3 is chemical engineering is responsible for fulfilling everyday needs such as clothes, food, and energy. These are the examples of chemical engineering products. So first is fertilizer, second plastics, third construction, fourth electronics, fifth is petrochemicals, sixth is gasoline, then polymers, food, pharmaceuticals, batteries, antibiotics, electricity, medications, sanitations, detergents, and detergents. So this is genetic engineering. So genetic engineering, also called as genetic modification. It is a process that is used in laboratory-based technologies to alter the DNA makeup of organism, or genetic engineering used to manipulate the DNA makeup of an organism. Example of genetic engineering is yung GMOs or genetically modified organisms, mga plants. Then the basic principle of this is to alter existing organisms by slightly modifying their genetic makeup, which involves using the recombinant DNA. So what is recombinant DNA technology? Please read. Rosemary. Okay, thank you. So, recombinant DNA technology is a technology used to producing artificial DNA through the combination of different genetic materials. This picture shows how genetic engineering or how recombinant DNA technology modify or alter the genetic makeup of an organism. So, plasmid. So what is plasmid? It is a small circular DNA molecule found in bacterial and some other microscopic organisms. Physically, separates from chromosomal DNA and replicate independently. So this circular DNA is called plasmid, and this is called a bacterial DNA. So what is restriction enzymes. So, restriction enzymes used to cut DNA into smaller fragments. The cuts are always made a specific nucleotide sequence. Then, different restriction enzymes recognize the cut different DNA sequences. It serves as a scissor. And what is DNA ligase? DNA ligase is used to form glue, or it is a fragment of DNA it is used to form fragments of DNA together to form new DNA strands during DNA replication and recombination. It has critical role in DNA repair in all organisms. Types of restriction enzyme. There are two types of restriction enzyme, the endonuclease and exonuclease. So endonuclease cut within the DNA strand and cut the DNA into specific points. The specific site is called restriction site. Exonuclease is to remove the nucleotides from the end of the strand. We have 5 processes of recombinant DNA technology. First one is isolation of genetic material. Second, cutting the gene and at the recognition sites. Then amplifying the gene copies through polymerase chain reaction and ligation of DNA molecules. Then insertion of recombinant DNA into host. Class, do you know what is GMO? Yes, Rusmay? Okay, thank you. GMO is genetically modified organism. Who among you here can distinguish these GMO products? Yes, Jikimaya. Okay, very good. It is a corn. How about this? Yes. Noramin? Oh, it is not a potato. It is a papaya. Okay, all of these are the examples of genetically modified organisms. So, okay class, since we already discussed GMOs or GMO products, for your assignment, please at least 10 GMO products that you can find at your home and pass it next meeting. Okay, but before we end our discussion class, please pick up some pieces of paper under your chairs and arrange it properly. Goodbye class. Goodbye sir. Okay, thank you.